Okay, thank you for your time. So, for today, I'll be speaking about stem cells. So, what are stem cells? Stem cells are a part of the human body that is capable of self-renewal, tissue repair, differentiation, and division into different cell lineage. So, there are three types. Next slide, please. Next slide. Thank you. So as you can see from the screen, there are three main types: embryonic stem cells, adult stem cells, and induced pluripotent stem cells. Embryonic stem cells are basically derived from inner cell mass of the blastocysts. Adult stem cells are mostly obtained from adult tissue. Multipotent or adult stem cells include mesenchymal stem cells. iPSCs are obtained from adult cells which can be reprogrammed like embryonic stem cells. All right. So next slide. So I'm going to talk a bit on each different type of stem cells, just to give you an idea. Adult stem cells are undifferentiated cells found among tissues or organs. They can renew itself, yield some or all the most specialized, all the major specialized cell type in a tissue or organ. The primary roles of stem cell is to maintain, repair the tissue they are formed. Next slide, please. Next slide. Embryonic stem cells are mainly derived from blastocysts. These cells can be derived in a lab. What you do is you can add various growth factors to stimulate the stem cells to generate different types in the body. Next slide, please. Okay, this is one of the key things in Asia Pacific. iPSCs, how are they formed? iPSCs in general in the lab can be taken from skin or blood cells, reprogrammed using OX-SOX, and to make them behave like stem cells. They are pluripotent, meaning they can change into different type of cells. There are several advantages of using these kind of cells for therapeutic purposes. They are ethical, and they can basically change into different type of cells. All right. So next slide, please. So for this is one of the key things I would like to emphasize. What are naive stem cells? A pluripotent stem cells basically has the capacity to develop into a cell type of any organism. However, there are two distinct stages of pluripotency: naive and prime. For the purpose of this slide, we will focus on naive. Naive stem cells are derived from epiblast cells of the pre-implantation of embryo, while prime cells are derived from post-implantation embryo. Although both naive and prime cells are able to self-renew indefinitely, express OCT4, SOX2, and differentiate down three germ lines, they are molecular and functionally distinct. So one of the companies we represent is Minmera. So next slide, please. So as you can see from this slide, we have different products in a kit to help harvest naive stem cells, all right? So for more details, you can actually contact our company directly. We will have a separate seminar to explain the science, all right? So yeah, this is a look of what we have here, okay? So we can jump to slide number 11. Yes, this one. Thank you. So what you have here, essentially, it's an enhanced feeder-free system from AlphaSTEM. So it basically gives you a higher improved self-growth rate. It also is a lot more easy to scale up, a lot more efficient in getting the number of cell types. So we have done a 3 comparison: feeder-based, one with matrix gel, and one using our system. All right. So next slide, please. So essentially, if we can see the images, it basically shows how much more cells we can get on a naive state using our system. We have tested up to more than 17 passages within a normal karyotype. Next slide, please. Okay, using our system, it has enhanced purity and cell function. It's a lot more scalable, a lot easier to automate. So this system, again, if you have any inquiries, please send it send to us. We have a lot more technical data we can email to you. Next slide, please. Okay, next slide. Okay. So in a nutshell, this is the system benefits that we have here. So we do not, with this system, has superior differentiation potential, a lot scalable, and expansion is a lot faster. All right. Next slide, please. All right. So on the next topic, MSC, this is one of my favorite topics, mainly because we have a lot of investor interest in several clients in Asia Pacific. Stem cells, MSCs can be from adipose tissue, bone, blood, and they are multipotent. They can self-renew. So right now for this particular slide, we will focus on a Korean company called Xcell. Next slide, please. So what are MSCs? MSCs basically are stem cell capable of differentiating into non-mesenchymal lineages, neural cells, endothelial, astrocyte, cardiomonocyte, etc. So it is very, very powerful tool in regenerative medicine and tissue repair. So these days, there's a lot of heavy interest in MSC. Maybe about 5 years ago, there was a lot of interest in CAR-T, NKT, but MSC is getting its interest in Asia Pacific. So it has a very unique ability to smart bomb and target site of injury, tumors, microenvironments. So this serves as a very excellent delivery system for the various therapeutic that people are developing, right? Next slide, please. So current tools, as you can see from this slide, essentially we have shown MSC's auto-homing to the site of injury. We have shown that it has been good to use as a delivery system for gene therapy as well. All right. Next slide, please. So MSCs typically are expanded in vivo before using in clinical application. So the quality of the media, the way you culture is actually quite important, how you use it for therapeutic purposes. So for this particular slide, we'll talk about the chemically defined and serum-free media for MSCs. All right. Next slide, please. So essentially, we represent a Korean company called Xcell. They developed their own range of serum-free and chemically defined media. So just give me a second. All right. So why do we choose this company? Essentially, they have run a lot of comparison versus leading players. Their cells are a lot more reproducible. The cells also retain a functionality, and the media is a lot cost effective when you factor in the number of cells you can produce. All right, next slide. So to highlight or educate a bit, there are a lot of competitors out there with different terminology. What is serum-free? What is chemically defined? Serum-free is basically products which do not have plasma or serum as an ingredient, but they may contain albumin or a bit of transferrin inside the media as well. Commonly, a lot of these media actually have human platelet lysate, all right? So what is chemically defined? Chemically defined means all the product itself, the ingredients have been chemically defined. There's mainly a compositions of amino acids, salts, vitamins, things that you can qualify. All right, next slide, please. So what is this CellCor SFD MSC? It's a serum-free MSC developed by the Korean company Xcell. It has shown to have superior growth rate compared to the competitors, and it has went up to 15 passages. Most companies try up to 7 to 10 passages. Next slide, please. I think I'm running out of time. So this is serum-free defined MSC. So this again is another formula designed to grow MSCs in a serum-free environment. So again, we have done tests to show the surface markers, the differentiation capacity of using this media. Next slide, please. Next slide, we can skip this. Yeah, all these are data showing how our MSCs perform using our media. Next slide, please. All right, so lastly, in the market right now, there are still clients using feeder calf serum to grow MSCs. So this in therapeutic purposes, the US FDA allowed 2% FBS in your media to for therapeutic purposes. So we represent this company called BioWest, which actually they provide high quality serum, which is traceable down to the point of slaughter, where the cattle has derived from the where the serum has been derived from. So this is very important for therapeutic purposes because a lot of markets products are just given a COA, but for us, we can give you the vet health certificate of origin, and the COA. This basically allows you to qualify the serum down to its source of material. Yeah. With that, this concludes the end of my presentation. Yeah, you guys can contact us directly if you think of any questions later. Thank you.